Hi, I am Renata Melo. I'm a master's degree student in the Cytogenetics and Mutagenics lab of the Department of Genetics of the School of Medicine of the University of São Paulo. I am under Dr. Elza Chiemi Sakamoto Ojo's supervision, and I'll be presenting my work, Potential of Novel Acetylcolinesterase Inhibitors TAHB3 and TA8Amino to protect BC12 differentiated cells against oxidative damage. Alzheimer's disease is an age-related neurodegenerative disorder that compromises cognitive functions and memory, and it affects more than 50 million people around the world. The main cause of cognitive decline in Alzheimer's disease is neuronal death that can be developed by many complex factors as oxidative stress and reduced levels of acetylcholine. Currently, the available drugs to treat Alzheimer's disease are acetylcholinesterase inhibitors that are used to treat patients with mild and moderate Alzheimer's disease stages. Therefore, there is a need of improvements to achieve higher efficacy and minimum side effects in the treatments of Alzheimer's disease. In this context, the hypothesis of the work was that the new acetylcholinesterase inhibitors TAHB3 and TA8Amino synthesized as donepezil-tecrine hybrid molecules may protect neurons from induced oxidative damage. We aimed to test the new hybrid compounds in mature neurons, which were differentiated from PC12 cells, and carried out neuroprotective assays to study the effects of the compounds against oxidative damage induced by hydrogen peroxide compared to the original compounds. In order to do that, the cells were differentiated for 7 days using neuron growth factor, changing the differentiation median in each 48 hours. After the differentiation, the cells were exposed to the drugs in their IC50 concentration for 24 hours, and then exposed to the hydrogen peroxide to induce oxidative damage for 2 hours. After that, they recovered for 48 hours, and then XTT assays were performed to analyze the cell viability. The results of neurodifferentiation were successful, were considered neurodifferentiated cells, the ones with at least one urite with twice the length of the cellular body diameter. After the treatment, a high percentage of differentiation was achieved. Regarding cytotoxicity, the results were positive because the compounds did not exhibit cytotoxic effects and presented high cell viability than the original compounds. As neuroprotection, until now, just one experiment was performed, but the results are promising because the hybrid compounds presented higher viability than the hydrogen peroxide alone, and similar effects were observed in previous studies with different cell lineages. Therefore, we can conclude that the results are positive and demonstrate promising neuroprotective effects of the novel hybrid compounds against oxidative damage. Additional experiments are currently in development to confirm the results and also to study other parameters as psychokinetics, cell death induction, and DNA damage, aiming to characterize the biological effects of the compounds and to confirm their neuroprotective properties. Thanks for watching.